Graduates. Dr. Gupta.
Thank you, Dr. Koslov. And thank you, Dr. Sundram, for the kind invitation. So lung cancer is actually the leading cause of cancer deaths around the world. And it is also the most prevalent cause of cancer in Malaysia itself. So lung cancer can be categorized by cell type into non-small cell lung cancer, which comprises about 87% of all lung cancer cases. And included in this are three different types: the adenocarcinoma, squamous cell carcinoma, and the large cell carcinoma. In addition, there is a small cell lung cancer, which comprises about 13% of all lung cancer cases. The prognosis for lung cancer is usually quite poor for patients, since the survival rate, the 5-year survival rate, is only about 15%. So looking at the different chemotherapy regimens which are available for lung cancer patients today, this was a study done earlier looking at the effect of cisplatin, the current most popularly used drug, with different agents on lung cancer patients. And essentially, what we see is that none of the 4 chemotherapy regimens offers any significant improvement over the others in the treatment of advanced non-small cell lung cancer. So traditionally, the cancer therapy was designed to interact, or most of the drugs which were used against cancer were designed to interact with general cell features, so that in that case, the cancer cells as well as the normal cells were usually killed. And that led to a very high toxicity and very high side effects. Nowadays, and more currently, there are targeted drug therapies which are looking to interfere with specific molecules involved in tumor growth and progression, and therefore they target cancer cells only, thereby decreasing some of the toxicity associated with these cancer drugs. Now, one of the specific molecules which we'll be discussing today is the Notch receptor and ligands as drug targets. And they have been, the Notch receptors and ligands have been implicated to a great extent in different cancer types, proliferation, differentiation, as well as in the increase in apoptosis. And they normally function by regulating different gene products, such as HES1, BCLX2, survivin, which are involved in cell differentiation and apoptosis. This chart actually shows you the dysregulation of Notch1 in different cancer types, and there is 30% dysregulation of Notch1 activity in non-small cell lung cancer. Now, it has been seen in patient population that there is a significant impact on the prognosis of lung cancer patients with decrease in Notch1 regulation. So there is a 69% improvement in the 5-year survival rate when Notch1 is suppressed. Therefore, Notch1 inhibition or Notch inhibition has become the current target for cancer therapy in non-small cell lung cancer. The key enzyme which is involved in the Notch1 pathway is the gamma-secretase enzyme. So pharmaceutical companies around the world are actually looking to develop synthesized drugs or chemical compounds which would help to decrease the activity of Notch1 or the gamma-secretase. And these are, however, most of these gamma-secretase inhibitors have failed clinical trials because of various levels of toxicity. So the idea is then to look for minimally toxic inhibitors of Notch1 signaling. In my lab, I've been working with various dietary bioactive components such as garcinol, curcumin, and now I've turned my attention to tocotrienols. So basically, our current understanding is, or we are looking to see whether or not these bioactive compounds have any synergistic effects with the current available drugs. Can they act as a part of an adjuvant therapy? So tocotrienols, the anti-cancer effect of, has been shown in various cancer cell types. And I won't dwell too much on it because we got an excellent talk in the morning by Dr. Sylvester on breast cancer and its effects, tocotrienols and its effects with breast cancer. So our central hypothesis is that delta-tocotrienol will behave as a therapeutic micronutrient in non-small cell lung cancer via inhibition of Notch1 signaling, and perhaps it may be used as a sensitizer to Cisplatin, the current chemotherapy available. So the first question that we like to look at is, will delta-tocotrienol show anti-cancer activity against non-small cell lung cancer adenocarcinoma cells? And further, if so, if it does show that decrease in activity, does it modulate it through the Notch1 signaling pathways? This work was published earlier in journal Cellular Biochemistry. So the slide here, we look at the effect of delta-tocotrienol on cell inhibition or cell proliferation. So you can see that in the two adenocarcinoma cell lines that we've selected, the A549 and the H1299, both show a decrease, a dose-dependent decrease in cell proliferation upon addition of delta-tocotrienol. And this was done by the MTS assay. This was further confirmed by the clonogenic assay, which pictorially you can see that the colonies formed with addition of delta-tocotrienol in both cell lines is significantly decreased as compared to the control cancer cells. Further, because cell proliferation can be accompanied by increases in apoptotic cells, we wanted to look at this effect as well. So here we see that there is a dose-dependent increase in apoptotic cells due to delta-tocotrienol intervention in both the adenocarcinoma cell lines. This assay done by histone DNA ELISA was further confirmed by NX and V staining, and we see that there is a significant increase in the number of apoptotic cells present, 58 versus 5% compared to the controls, in the delta-tocotrienol treated cells in both the cell lines. Also, because apoptosis is related or is related to cell cycle, we wanted to look at whether or not delta-tocotrienol affects the cell cycle at any checkpoint. And what we saw was that indeed it induces cell cycle arrest at the G0, G1 phase of the cell cycle. Now further going on to look at the effect of delta-tocotrienol on Notch1 regulation. If you now concentrate on panel 1, we see that there is a dose-dependent significant decrease in Notch1 activity, protein expression using the Western blot analysis, which shows that delta-tocotrienol affects the level of expression of Notch1, the protein level as well as the mRNA level, which is seen in panel C by real-time RT-PCR studies. And having seen that it affects the Notch1 regulation, we then went on to look at the downstream genes involved in the Notch1 pathway, and we saw that other proteins such as HES1, PARP, survivin were also affected by delta-tocotrienol intervention, both at the mRNA level and at the protein expression levels. Now, in addition to Notch1, there has also been a lot of recent literature regarding the implications of NF-kappa B, the signal transduction factor, in cancer progression. And also, in addition to that, there has been reports on the crosstalk between the Notch and NF-kappa B signaling. So in addition to Notch, we wanted to see whether or not delta-tocotrienol will affect NF-kappa B DNA binding activity. So as shown by the EMSA plot in panel A, you see that there is effective and significant decrease in the NF-kappa B DNA binding activity in a dose-dependent manner on addition of delta-tocotrienol. again in both cell lines. Further, when we look at the effect on the gene, downstream gene products of NF-kappa B signaling, the VEGF and MMP9, both of which are involved in metastasis of cancer, angiogenesis, and migration of cells, we saw that there was a dose-dependent decrease in the expression of both of these genes. Thereby, the implication is that NF-kappa B and its downstream genes have been inhibited by addition of delta-tocotrienol. This was further investigated by using the matrigel invasion assay, which again, you can see that the number of cells which have invaded or crossed over the boundary has decreased substantially and significantly in when you compare the tocotrienol treated cells with the control cells. And further, we looked at the wound healing assay, which was, again, it is basically done by creating a wound or with a clean pipette on these, on the optimally growing cells. And what happens is, if the control, if you look at the control cells, the wound gets healed, so the cells grow back into the wound which is created, whereas with delta-tocotrienol, the cells remain separate, which means that the cells were not invading as aggressively. So to summarize this portion of the talk, we saw that delta-tocotrienol inhibited Notch1 signaling via NF-kappa B inhibition, which led to a decrease in PARP, BCL, XL survivin, all involved in decrease in cell growth and an additional increase in apoptosis. Also, because of the decrease in VEGF and MMP9, we also related that to a decrease in invasion and migration of the non-small cell lung cancer cells. Now we have seen that microRNAs, which are small non-coding RNAs, tend to exhibit their function by blocking transcription of and binding to the messenger RNA assembly. And so we wanted to see whether Notch inhibition by delta-tocotrienol is somehow accompanied with alterations in the microRNA expression profile in the non-small cell lung cancer, cell lung cancer cells. So this plot is actually showing, so we did a microarray analysis in which we looked at some 80 plus different microRNAs, and the lines here, the boundary lines here, indicate a 4-fold increase in the microRNA expression. And from here, from the microRNA analysis, we saw that the expression of MIR34A, the microRNA 34A, was significantly increased with delta-tocotrienol treatment as compared to the control cells. In panel B, you see that addition of delta-tocotrienol over time increases the expression of MIR34A, and also we see that expression of MIR34A increases with a dose-dependent increase in delta-tocotrienol. So yes, delta-tocotrienol does increase or does induce the expression of MIR34A. Now, microRNAs can function both as oncogenes or oncomirs, that means tumor suppressors. So we wanted to see what the effects of microRNA 34A was with respect to cell proliferation. So in order to do that, we transfected cells with microRNA, and let me explain this a little further. So this bar here refers to the negative control. So the cells were transfected with a negative control for microRNA 34, which again, if you look at it, there is hardly any inhibition of cell proliferation. So they're pretty much the same as the control cells. Now, if you instead transfect with microRNA MIR34A, we see a decrease in the cell proliferation, and this was statistically significant. Now, in addition to that, we also transfected the cells with an antisense of microRNA. This is sort of like creating a knockout model for microRNA in the cells. So with that, with doing the knockout of microRNA 34A, again the inhibition has, and again the cell proliferation is not affected by in these cells. Now, addition of delta-tocotrienol, of course, decreases cell proliferation, and you see that when you add delta-tocotrienol to the knockout model, the cell proliferation is not decreased significantly. This was further confirmed by the clonogenic assay at the bottom. Again, you see a decrease in the number of colonies produced because of transfection with microRNA 34A or delta-tocotrienol. So, which shows that the MIR34A functions by decreasing cell proliferation in the cancer cell lines. Now, in addition to decreasing cell proliferation, it also showed that it induces apoptotic effects when MIR34 is re-expressed in these lines, so in these cell lines. So again, the transfected cell lines with MIR34A show an increase in apoptosis, which was also seen with delta-tocotrienol. And when you have the knockout model, the effect of delta-tocotrienol is decreased. This was further seen in the bottom slides with annexin-5 staining followed by flow cytometry, showing you an increase in the apoptotic cells with addition of either MIR34A or delta-tocotrienol. Again, in addition to the changes in apoptosis, we saw changes in the G0, G1 cell arrest by 34A and delta-tocotrienol. So looking back at the Notch1 expression and the target genes, we again see that if you look at the Western blot here, so we see that the Notch1 expression is lowered in both the transfected cell lines transfected with 34A and delta-tocotrienol, but the effect of delta-tocotrienol gets reduced if you use the knockout model, that is the lines where the cell lines where 34A has been negated. And this was seen in the expression of Notch1 as also the gene products HES1, cyclin D1, and survivin. The lower panel actually looks at the invasive ability of the MIR34A, and it follows the same trend as the cells being treated with delta-tocotrienol. So to summarize this portion of the work, what we saw was that the effects, what we consider is now that the effects of delta-tocotrienol on Notch1 via NF-kappa B signaling and the downstream signaling occurs via upregulation of MIR34A. So MIR34A seems to be integral to the effect of delta-tocotrienol in non-small cell lung cancer cell lines. So now we turn tables a little, turn direction a little bit, and now we want to see whether there are any beneficial effects of using the delta-tocotrienol in suboptimal concentrations, that lower than the optimal concentration, in conjunction with Cisplatin, which is the current therapeutic drug used for lung cancer patients. So this work has also been published and was done in 2012. So the panels A and B here show the anti-proliferative effects of delta-tocotrienol and the combination on the non-small cell lung cancer cell lines. And we see that the combination of delta-tocotrienol and Cisplatin is more effective, considerably and significantly more effective in lowering the cell proliferation in both adenocarcinoma cell lines, which was again validated using the colony formation assay. In addition to the synergistic effect in lowering cell proliferation, we also saw synergism in increasing apoptosis. So here is the effect of the apoptotic effect of delta-tocotrienol alone. and then followed by that of cisplatin. And if you look at the two in combination, you see a tremendous increase in apoptotic activity. And again, this was further confirmed by flow cytometry. So going back to our initial thought that delta-tocotrienol downregulates Notch1 expression, we wanted to see whether the combination again is better than either of the two agents alone. So here again, we see that the effect of the two agents in combination, delta-tocotrienol and cisplatin, as you can see over here, for example, at the Notch1 signaling, you see that the delta-tocotrienol alone, and cisplatin actually doesn't do too much for Notch. The combination, however, is more effective than either of the two. And this followed for both the cell lines and the downstream gene products of the Notch1 pathway. The interesting aspect came up here when we started looking at the effect of the combination of delta-tocotrienol and cisplatin on the NF-kappa B activity. So here, we see that, as we saw before, that when you treat the non-small cell lung cancer cells with delta-tocotrienol, we see a decrease in NF-kappa B DNA binding activity. However, when you treat the cells with cisplatin, NF-kappa B activity, DNA binding activity, is not reduced. In fact, certain systems show an increase in the NF-kappa B activity with addition of cisplatin. However, when you put the two in combination, they do show a decrease in NF-kappa B activity compared to the control cells and compared to the cells treated with cisplatin. In addition, you also see a decrease in the downstream signal molecules, VEGF and MMP9, which is considerably better in the combination of the two agents as compared to either of the agent alone. Similarly, and it follows that when you look at the cell invasion assay, we see a significant decrease in the cell numbers which are showing invasion in the combination treatment as compared to cisplatin alone or delta-tocotrienol alone. Again, in the wound healing assay, you see that the combination of delta-tocotrienol and cisplatin tend to keep the wound apart, meaning that the combination is much more effective in reducing migration, which leads to metastasis or spread of cancer, as compared to the control cells or the cells treated with pure delta-tocotrienol or pure cisplatin. So to summarize this, we see that there is a combinatorial effect of delta-tocotrienol and cisplatin in non-small cell lung cancer cells. What was interesting to me was that cisplatin actually increased NF-kappa B, and looking into literature, we found that because NF-kappa B increases inflammation along with other activating other pathways, an increase in NF-kappa B is often related to increase in drug resistance or increase in drug sensitivity. So it would be interesting to use the two in combination to see whether or not the delta-tocotrienol, by upregulating miR34A, could then lead to a decrease in NF-kappa B activity, leading further to sustained inhibition and migration, inhibition of cells, migration and invasion of the non-small cell lung cancer cells, along with a decrease in the genes regulating cell proliferation, apoptosis, migration, and invasion. So to summarize what we have shown here, what I've shown here is that our data shows that delta-tocotrienol inhibits non-small cell lung cancer cell proliferation, invasion, induces apoptosis by downregulation of Notch1 signaling pathway, as also of the NF-kappa B DNA binding activity. And it seems that delta-tocotrienol is a non-toxic activator of MIR34A, which itself is acting as a tumor suppressor. So by inducing it, it is suppressing some of the properties of tumor progression. In addition, we also saw that there was at least an additive, if not synergistic effect, in combination with Cisplatin, the current drug for non-small cell lung cancer, and it functions by downregulating both Notch1 and the NF-kappa B pathways. So given these factors, it can be considered that the combination of Cisplatin and Delta-tocotrienol can be a potential therapeutic regimen which could be used in further studies. So the data that I've presented so far has been published, and currently we are looking to see whether or not we can transform or translate the data from our in vitro models in the cell culture to Delta-tocotrienol's performance in vivo. So when we were thinking about the transformation, we realized that we cannot quite do a lot of studies with the pure Delta-tocotrienol, and we needed to go to the commercially available tocotrienol-rich capsules. And essentially at this point, and I'm not going to show the data here, but at this point we have basically repeated all the studies from our pure Delta-tocotrienol to the tocotrienol-rich capsules, and seen that the tocotrienol-rich capsules, which are, of course, a mixture of different tocotrienols, actually they have higher proportions of the gamma and alpha tocotrienols, and they are also very effective in reducing cell proliferation and increasing apoptosis in the non-small cell lung cancer cells. In addition to that, we have also extended our study from primarily the adenocarcinoma cell lines, which we were using for the pure isomer Delta-tocotrienol, to the squamous cell line. And the reason we did that was because patients which have additional non-small cell lung cancer in the form of squamous cell lung cancer have a poorer prognosis. And most current therapies do not function as well on the squamous cell lines. And by including squamous cell lines with the commercially available capsules, we've actually expanded their potential. And so this work is now being conducted in collaboration with my team at Karmanos Cancer Center, part of Wayne State University, and we are looking forward to doing a phase 1 human trial. At this point, I would like to thank you all for your attention. I would like to thank Wayne State University for the funding for this project. Carotino provided us with the tocotrienol capsules. These are the same capsules which were used by Dr. Khosla as well for his renal work. And the pure Delta-tocotrienol was provided by American River Nutrition Incorporated. I would like to thank my wonderful students, and Xiaoming Ji was, this project was really his PhD work, and he's now a postdoctoral fellow in another lab. And I would thank MPOC for the very kind invitation to come to Malaysia. Thank you.